So, in the world of microbiology, you've got this really unique challenge. How do you identify a suspect you can't even see? A suspect you can't exactly sit down and question? Well, in this explainer, we're going to find out. It turns out you do it just like a detective. You follow the evidence they leave behind, cracking the case by uncovering their unique metabolic fingerprint. And that very question is what turns the microbiology lab into a kind of detective's office. We're going to follow the clues and see how scientists use a series of really clever tests to basically force a microscopic suspect to confess its identity. You see, instead of a police lineup, scientists use a biochemical one. They put the bacteria up against different challenges, like here are some different food sources, or let's see how you do in this environment. And how the bacteria responds? Well, that's a clue. You start building a profile, and that profile is absolutely crucial. I mean, it's how doctors figure out the right antibiotic for an infection, or how investigators can trace the source of a foodborne illness. It's all about making that final positive ID. So you can almost think of us as forensic accountants for the microbial world. We're going to be tracking how different bacteria process carbohydrates, what they eat, and the chemical breadcrumbs they leave behind. This metabolic trail is exactly what sets one apart from another. Okay, our first line of questioning. It's pretty simple, but it is absolutely fundamental. We just need to know, does our little suspect need oxygen to break down its food, or can it get the job done without it? This is where the oxidation fermentation test, or just OF test for short, comes in. So bacteria basically have two main strategies. You've got oxidation, which you can think of as a slow, really efficient burn that needs oxygen to work. Then there's fermentation. This is a super rapid process that doesn't need oxygen at all, and it produces totally different stuff like acid. This fundamental difference? Well, that's the key we're going to use to pick this lock. So how do we get our answer? We run a pretty slick, controlled experiment. We take our suspect bacteria and put them into two test tubes. One tube is left open to the air, so oxygen can get in, but the other one we seal with a layer of mineral oil, which completely blocks out the oxygen. This little setup basically forces any fermenters to show their true colors. And this is where we get the confession. The test medium has a pH indicator in it. So when the bacteria break down the sugar and produce acid, that indicator makes the whole tube turn from green to yellow. Just by looking at which tube changed color, the one with oxygen or the one without, we can figure out the bacteria's entire metabolic strategy. So here it is, our detective's codebook for the OF test. If only the unsealed, oxygen-filled tube turns yellow, we've got an oxidizer. If both tubes go yellow, then it's a fermenter. And hey, if neither one changes color, well, that just means our suspect can't metabolize that sugar at all. And just like that, clue number 1 is officially in the logbook. All right, so now that we know how our suspect breathes, we got to dig a little deeper into its diet. Our next clue is all about finding out what specific things it eats. Can it ferment sugars like glucose or maybe lactose? And the follow-up question is, when it does that, does it, produce gas? This is where the phenol red broth test comes into play. The PR broth is our next interrogation tool. Inside this nutrient broth, you've got 3 key things: a specific sugar we want to test, a pH indicator called phenol red, and this clever little inverted vial called a derm tube, which is perfectly designed to trap any gas bubbles the bacteria might make. And reading the result is actually super straightforward. If the broth turns yellow, it means acid was made, so fermentation definitely happened. A bubble in that tiny derm tube tells us that gas was also produced. But if the broth stays red or even turns a deep pink color, that means no fermentation. The bacteria probably just used a different nutrient source, making alkaline waste instead of acid. And of course, just like real detectives, microbiologists have a shorthand: A/G for acid and gas, A/- for just acid, and K for an alkaline reaction. Every single detail helps build that unique profile of our suspect. Okay, on to our third and final clue. And for this one, we're getting really specific. See, it's not enough to know that acid was produced. Now we need to look at the exact chemical trail it left behind. And that is the job of the methyl red and Vosges-Proskauer test, or as everyone calls it, the MRVP test. This quote really sums it up perfectly. The MRVP test is actually 2 tests in one. The methyl red, or MR part, checks for something called mixed acid fermentation. That's a process that creates a whole bunch of strong, stable acids.
The Voges-Preskauer part, or VP, looks for a completely different pathway, one that ends up producing a more neutral thing called acetoin.
And the process for this, it's actually super elegant. Scientists grow the bacteria in just one MRV broth. After it sits and incubates for a while, they just split that one sample into two separate tubes. They add the MR test chemicals to one and the VP chemicals to the other. So from one sample, they get two incredibly powerful clues. So what's the real difference here? Well, an MR-positive organism produces so much stable acid that it just keeps the pH super low. Think of it like leaving a permanent acid stain at the crime scene. A VP-positive organism, on the other hand, it ferments glucose, but then it quickly converts those acids into acetone. It's basically cleaning up its own acidic trail. And here it is, the final confession. After adding the right chemicals to each of those tubes, a red color means a positive result. If you see red in the MR tube, that confirms mixed acid fermentation. Red in the VP tube, that confirms acetoin production. The suspect has just revealed its specific MO.
All right, we've gathered all our clues. We know how our bacteria breathes, we know which sugars it eats, and now we know the precise chemical trail it leaves behind. It's time. Time to assemble the evidence and put a name to our culprit.
So let's look at the profile for a very well-known suspect, Escherichia coli, better known as E. coli. From the OF test, we know it's fermentative. From the PR broth, we know it ferments glucose, making both acid and gas. And from the MRVP test, we see it follows that mixed acid pathway but does not produce acetone. This exact combination of results, that is its unique, undeniable metabolic fingerprint. And that is the key to this whole thing. It's not one single test that cracks the case, no way. It's the unique pattern of results across all these different tests that let scientists build that definitive profile and tell one microscopic organism apart from thousands of others. It's just a powerful reminder that this invisible world all around us is just packed with information. Every single organism leaves a trail, a unique chemical signature. The whole trick is just knowing how to look for it. Which kind of makes you wonder, doesn't it? What other secrets are hiding in plain sight, just waiting for the right test to bring them to light?